Well, good evening, ladies and gentlemen. It is my great pleasure to invite you to this evening. I've been given the pleasure on behalf of the Czech Liaison Office for Education and Research to welcome you here today. The mission of our office, as some of you know, is among others to showcase Czech scientific excellence. And we're very happy that we get to do that with the hosts of this event, the Prague House, along with the representation of Southern Moravia to the EU, as well as Czech Center Brussels. With the planning of this event, we were fairly lucky because, as some of you who follow scientific news know, the UK has introduced the world's first legislature very recently, allowing the use of CRISPR for the treatment of sickle cells and sickle cell disease and beta thalassemia. Well, this revolutionary decision opens up new doors for new solutions, but also raises a lot of fairly ambiguous ethical questions. We're very glad that Alžběta Ressnerová has accepted our invitation for tonight to help us sort of navigate the ethical minefield of gene editing. And also to present us her own research focusing on gene therapy as well as nanorobots. But I will let Olga Maximová, today's evening moderator, to tell you a little bit more about Alžběta in a second. Some of you might remember Olga from our previous edition of Edu Café, where she focused on the women in STEM fields discussion. And she is also the head of development of CEITEC, the non-profit organization in the Czech Republic, which aims to make IT education more accessible to disadvantaged women in the Czech Republic. And she's also an experienced moderator. So, Olga, the floor is yours. I'll leave you to it. Have a pleasant evening.
David has just said everything I wanted to say, so there's not much for me to. But we didn't know where to sit, David. Are there designated places for us to? Okay, I look better from the left side, so. You can you can sit wherever you like. Anyway, ladies and gentlemen, it's my pleasure to be here. I'm really sorry for the wait, but I was moderating another discussion about gaslighting and the effect on young people in today's world. Maybe the young gentleman would like to tell me more about it later. So I'm sorry we're starting maybe a bit later, but I would like to welcome a very extraordinary person here tonight, Alžběta Ressnerová. She is a Czech scientist, researcher. How do you reference yourself? A scientist, a researcher? A scientist sounds better. I really like it. Alžběta's work at the intersection of molecular medicine and nanotechnology has been pushing the boundaries of what is possible in biological applications. I will tell you a little bit more about her background, but then it would be good if we could also explore that. Alžběta finished her master's degree at the Humboldt University in Berlin in molecular medicine before returning to Czech Republic. But now she works or researches at CEITEC, which is Central European Institute of Technology, where she is using nanorobots and nanotools for diverse biological applications. I can't wait to discover what it is. I don't know whether there are any laymen or laywomen in this area. Can you raise your hand if you really don't know what we're going to talk about tonight? Great. You are going to love it. You're going to discover a brand new world, because I did when I was researching and reading about the area, and I thought, like, oh my God, holy crap, that's such a shame that I never studied science. And how many of you are from the field? Are there any scientists here? Yeah? Anyone who's ever applied CRISPR? Oh yeah? I will remember you. Good. Good. I would like you, if you can, as we talk, as we discuss, please make note of your questions. It would be really good if we were an engaged audience. This isn't only about me facilitating and Alžběta talking about her research, but also you're here to find out what is happening in this field. But maybe we can take a seat now. Please, you look better from the... Right, you can sit there, that's fine. So, Alžběta, we will be talking about gene editing today. Yeah, try the mic.
Yeah, I think it works.
I never really thought that something like that was possible. Can we start, though, with something a bit different? Because you now focus on nanotechnology and nanorobots, right? And these two themes, nanorobots and nanotechnology and gene editing, don't have much in common. Is that right?
No, they don't have much in common. Yeah,
Okay.
That's true.
Okay, so can you tell us what you focus on now and a little bit about nanorobots?
So I'm basically trying to merge these two fields, field of nanotechnology and field of molecular medicine. What I'm trying to aim is to create a delivery platform for biologically active molecules. And these can be, like, you can count like chemotherapeutics as biologically active molecule, or it can be CRISPR for gene editing. So it's really important to have a vehicle for the therapy. So you cannot inject gene therapy in form of CRISPR just like that, intravenously. It's not going to do much. You need a vehicle that's going to deliver it to the target organ, to the target tissue, to the target cells. Also, we don't want therapies to cause side effects. Like, for example, chemotherapy has a lot of side effects. If you just inject it systemically, it's going to do a lot of trouble. So the idea behind merging nanotechnologies and medicine or biology is to create more targeted, more specific therapy that would be smarter, that would be more gentler, and that would be also efficient. So yeah, this is what I'm trying to do, to create these vehicles that deliver stuff in your body.
So the vehicles, the carts or the trolleys, are they nanorobots? Is this what we would refer to as nanorobots?
Well, not always. So the last time I actually worked with nanorobots is a few years ago. Right now I don't work with them currently, but I'm going to work with them in the future for sure, because I really have them close in my heart. I really love them. They're really cute.
They're cute.
And
how do they look, by the way?
Well,
are they something palpable? Can we see them, or are they more
you can see them under
microscope?
Like, yeah, microscope, but not a normal microscope. It has to be like, yeah. So nanorobots, many people think that they look like little men with like heads and antennas and stuff.
Why men? Why not women?
Oh, or little women.
That's my field, you know. So.
Of course. With like antennas and hands and things. And right now it's not true. They look like mini rockets or like mini Tic Tacs. Like the bonbon that you eat.
Tic-tac,
like,
yeah.
So, and they are fueled. They are somehow, like, they are moving. They're actively moving. And they can be fueled by various fuel sources. You can use, you can use like stuff like glucose, which is nice because we have a lot of glucose in our blood. So they could be powered by glucose in blood. Or you can use magnetic field, or you can even harness other cells and create nanorobots out of them, like, for example, sperms and so on. So it's really exciting. But back to your question, these vehicles that we talked about, they're not always motorized. You can also create smart nanoparticles, and they're not going to be motorized. They're going to be passive. But nanorobots, that's a new step. That's like an extra step. And nanorobots are very new in the nano family. They're pretty new. They're the youngest offspring, basically.
That's great. I wanted to ask you, so, this isn't a question that I have, it just occurred to me. It must be very expensive to design, create a nanorobot, something so small that can actually get into your body and fix a gene. Oh my God, like that must be very expensive. Can you maybe comment on that? Like how do they?
Well, I think science in general is expensive. It really doesn't matter that much what you do. It's all very expensive. And even myself, I can't really imagine how such a therapy, how expensive it would be if it was applied on humans. Right now, it has never been in like nanorobots. But yeah, it definitely is expensive, but so is many other things.
Fantastic. Okay, thank you very much. Can nanorobots be used for gene editing?
Well, right now It's more like a sci-fi, honestly. I think that in the future, yes.
But that's something you focus on, right? It's something that you
I
want
don't
to-
actually
no?
connect nanorobots and gene
Okay.
therapy. It's like two separate things. I'm more trying to create like passive nanovehicles right now for gene therapy, because nanorobots are so new that it's really hard to get them into clinic. You know, so it's like it's a basic research, and if you use nanoparticles, it's much easier to be more translational with it and maybe get it into a clinic one day. So yeah.
Yeah, we're gonna get to that with one of my questions. But I wanted to move to gene editing. You worked at the in the Institute of the Nobel Prize winner Jennifer Doudna and her colleague Emmanuelle Charpentier.
It's just Jennifer's institute.
Just Jennifer's, okay, at the University of California, Berkeley. Because you are the recipient of a very prestigious Fulbright Masaryk scholarship,
Yes.
fellowship, right? So can you tell us what you did at the institute, whether you worked directly with Jennifer, and then we will move a bit on. I would like to explore more about what her contribution was. What did she get the Nobel Prize for?
So I didn't work like under Jennifer. I did have several meetings with her, and I met her several times. I talked with her several times, but I didn't work under her. But the institute is absolutely amazing because she was able to put together several teams, several laboratories of amazing people who are all striving to make gene therapy accessible, which is something I really love, and it's very unique. So her vision is to make gene therapy accessible to everyone, not to be a thing for rich. You know, so it's accessible in poorer countries to underrepresented communities and historically oppressed communities as well. So I really like this noble cause, which is, I think, very rare to see. And those scientists that work there are like the best in the field, in the world. So it was a huge privilege for me to be able to work there.
Yeah, it's a huge prestige. And can you tell us a little bit more about what she got the award for, the Nobel Prize?
So Jennifer Doudna got the Nobel Prize in 2020 together with Emmanuelle Charpentier, and they got the Nobel Prize for discovery of CRISPR. And for discovery of CRISPR as a gene editing tool. And that's very important to say, because CRISPR was first discovered as bacterial immunity, and I can elaborate a little bit more on that. And actually, the first paper that kick-started this whole thing and the Nobel Prize was a paper in 2012 in Science. And this paper talked about CRISPR as a bacterial immunity and outlined the idea that it could be used for gene editing. And the first author on this paper is Martin Jinek, who is from Třinec, from Czech Republic, which is absolutely amazing that in the beginning of this huge discovery stand this Czech guy who is a brilliant scientist. And of course, the main authors also were Jennifer Doudna and Emmanuelle Charpentier.
Okay, really nice. I'm going to sidetrack just a little bit because you may have kids at home who are thinking about studying and improving their education. When we spoke, I mentioned the Fulbright Masaryk stipendium, the fellowship, which is a very prestigious one. I still think very highly of people who get that stipendium. When we talked, Alžběta, you mentioned that actually you didn't find it that difficult to receive it. Can you tell us more about it? Because
I didn't know you were going to talk about that.
Surprise! Maybe it will support people in applying for that. Some people maybe think, oh, I'm just an ordinary Czech. I'm not going to apply. I'm never going to get it. Tell us more.
It's a great thing to talk about, actually. I think, I said that it wasn't that hard, and the truth is that if you do extra stuff and you are interested throughout your life, it's not that hard to get such prestigious scholarship eventually, because you already did the hard work beforehand. And I took it as a, like a gift or something like that, for the hard work that I did previously. So, and also I realized that many Czech people tend to think really lowly of themselves. I don't know if that's a word, like small of themselves.
Underestimate themselves.
Underestimate themselves. And there is no reason for that. Like, we are smart and we have great minds in our country, and many people are so scared of disappointment, of being rejected, that they don't even try. And I actually thought it's going to be much harder to get the scholarship. I didn't think I'm gonna get it, and eventually it was fine. It was okay. Like, you know, if you have a good project and you have a good aim, and you say, like, why is it so important for you to do this research in the US? Like, why you can't do it in Czech Republic? And you know, if all this fits into their picture, you're good to go. Like, that's it.
And how did it change your, let's say, career or your future studies? How did it influence you?
Oh, it was a major career
A milestone.
point. Yeah.
Yeah. In what way?
Well, just the experience of working under such amazing researchers and working, with people who are just like the best in the field. This is what I wanted from that. I wanted to work with the people who are the best in the field, so I could learn from the best. And this is exactly what it delivered.
Yeah, very nice. Well, congratulations.
Thank you.
Alžběta, could you explain the concept of CRISPR and its functionality in altering the genetic sequence?
So it's really interesting because I don't know if you knew, but bacteria, certain types of bacteria have immune system. And we never learned that at school because we didn't know about it back then. But bacteria can get infected by viruses. And scientists found out that certain types of bacteria are immune to viruses. And they were wondering why. And they found out that those types of bacteria have a library in their genome. And in that library, they save bits and pieces of DNA of viruses that attacked them before, so they can remember them. So once a virus attacks this bacteria goes to its library, scans it, and asks, like, have I encountered this virus before? And if it has its sequence in the library, it's capable to go back to the virus and cut its genome and basically gets rid of it. So this is really interesting. This is how CRISPR was discovered. And then it was proposed that we might be able to engineer this whole system in a way that it can be used to cut human genes, to cut human genome precisely. And I wanted to explain it on a video that I like to use. So in every cell of your body, you have nucleus. There are some cells that don't have nucleus, but let's say in every cell. And in the nuclei, it's fine if you don't have it, it's okay. It's okay. You can do it without it. Yeah. So, and in the nucleus of your cells, there are chromosomes. And those chromosomes are made of DNA. And each cell of your body has the same set of chromosomes, has the same DNA. And this DNA is called genome. It's a set of all those genes that are in your body. And what CRISPR can do is that we can design so-called guide. It's a really tiny piece of RNA which is specific to specific sequence in your genome. And we know how our genome looks because it was sequenced. And so you can tailor this whole system in a way that it scans the genome and searches for the sequence in the genome that is compatible to this guide. And this guide is bound to Cas enzyme. And this enzyme acts as molecular scissors, basically, and cuts the DNA precisely. So you can basically like engineer this whole machinery in a way that you know where it cuts precisely. And even when I say precisely, we know the nucleotide where it cuts. So nucleotide is all those A, T, C, G that you have in your genomes, those letters. So we know exactly in the whole genome where the system is gonna cut. And this can be used in various ways. Like you can, for example, make the genome lose its function. Like when you have a gene lose its function, sorry. So when you have a gene that is like making some bad stuff, like causing a disease, or there is a mutation that is causing a disease, you can turn the whole thing off. Or you can, or
like what, for example? Could you give us an example?
oh, well, for example, you have certain types of cancer that where certain types of genes are turned on, and they are producing huge amounts of protein that is not physiological. So that's causing the cancer. So you can turn this off. Or for example, you can replace a whole gene. If you have a gene that is not functioning, with CRISPR, you can replace it with a functional one. Or you can insert a new sequence inside. And you can also regulate genes with it and so on. So there is a lot of stuff that you can do with CRISPR. And this is what is so unique about it, because we had gene therapy before, but it was all very random. We basically took virus, we gutted the virus, used just the envelope, and then just inserted the gene inside and let the virus infect the cells. And you had no idea where it's going to insert the DNA. And what happened back then, back in the days in 90s, was that once this went to clinical trials, those patients started having leukemia because those genes that were just like put into a virus and they let them infect the cells, they were just inserting the genes absolutely randomly in the genome. And sometimes they just inserted them in the wrong place and cancer happened. So right now we have absolute control of where we are inserting stuff or what we do with it.
But let me just understand, you insert the stuff in labs, right? There has been a country which has greenlit this therapy to be used in clinical trial. Is that how we would use it?
It has been already in clinical trial. This is approval for use in clinic.
Oh, right. So which country was it?
It was the UK. And I think it was a week ago or something, like 2 weeks ago. It's really recent news. And it's really amazing because it's just 11 years since the paper from Martinique that started this whole thing.
So what does it mean in practice? I will go...
It's really fast.
The NHS is going to cover it.
I don't know about that.
But if I go to a doctor in London, they will inject it in me just right there on a spot.
No, not like that.
Okay.
This is what we are actually trying to do. So it's possible to inject it. But right now it's called ex vivo. So you need to take the cells outside of the patient. So they take stem cells. They take them out of the patient. They correct the stem cells. And then they put them back. So it's still tedious, it's still a burden on the patient, but it's curative, which means you change the person's life forever. We never had curative treatment for sickle cell disease, and it's a horrible disease. So those people with sickle cell...
and that's usually in developing countries, is that right?
It's not about developing countries, it's a thing of black people, so it doesn't matter where they are. So it's tied to them. And the reason why we see sickle cell in black population is because it's evolutionary adaptation to malaria. So people who carry the sickle cell trait can't get malaria. But once you inherit those both mutations from both of your parents, you get sickle cell disease. Which is horrible, and like people die at 48 years old in horrible pains. So it's a debilitating disease, and the fact that right now we already have CRISPR in clinic in the UK is a huge hope for millions of people.
That's great. I wanted to also ask you, you've mentioned something. How does the process differ when you apply the CRISPR to an individual like myself, for example, and to embryo?
Yeah, there is a huge difference because once you edit an adult, when the adult dies, their offsprings are not going to carry the edit, the thing that you changed. But once you CRISPR an embryo, you're basically creating a new line of humans. So if you edit an embryo, you edit every cell in the body, basically, of the future human, which means also the sperm or the egg. So once this human has children, there is a huge probability that this edit is going to be inherited. So you're basically, creating a new line of humans, of edited people.
Do you guys have any questions after what you have just heard? Actually, I think it's a good time to take questions. I still have a few, but I would like to pause here a bit and give you a chance to maybe clarify something. You know, I studied literature, so some of my questions might not be exactly what you would ask. But please, let's do. David, will you help us with that?
Sure thing.
Thank you so much. Anyone a question? The gentleman who's a pro in CRISPR.
He
Do you have
probably
any
doesn't have
questions?
questions because he is a pro.
I'm not a pro, but I'm absolutely fascinated by your story.
Thank you.
It's really nice to hear the enthusiasm and the passion for what you're doing, and that you're actually decided to come here and also talk about that. And I think it's just a wonderful thing. So
Yeah, thank you. We can
I
talk
used to
later.
do science. I used to do science in plant research, where we used, well, colleagues of mine still do the editing and stuff like that. I've never really been very around, so I've left the science at the time when the CRISPR was being discovered by Jane. But it's still amazing piece of work, and I'm just following on the development of the technology that, as you just said, the speed with which it was
It's
sort of
amazing.
deployed is amazing. The fact that the Nobel Prize was awarded within just a couple of years, it's a huge recognition. And I think the promise that this technology can bring is also
Yes.
marvelous. Yet, I mean, there are also
Yeah.
some sort of a
We are going
the
to talk about
dark
them.
sides of that.
About the dark
Thanks a lot.
side. Thank you for the addition.
Anyone else? Oh.
And then the lady
I
here.
have a technical question. So you talked about how the CRISPR technology enables to cut the sequence at a particular place, but I didn't get to understand how do you recombine, or how do you make the ADN recombine? How do you get rid of that sequence? How do you evacuate it? What's happening after cutting, basically?
The cell takes care of it. So basically, it's not allowed in a cell for any floating DNA to survive for a long time. So once you cut it out, the cell takes care of it and destroys it. For the you mean the floating piece, right? Yeah. Mm-hmm. The cell machinery takes care of it. It's not going to be there anymore.
It's not like there's a waste of DNA floating in us.
No.
Good.
And I think there was one more in front.
Yeah, the lady here.
So thank you for the presentation of your work. It's really fascinating. I would like to ask, how are these tests provided? You mentioned the clinical tests are started just a few weeks ago. So could you please a bit explain how it's provided? Because for someone who has not scientific education at high level, it's maybe quite complicated to imagine how it all works and how you want to put it in practice.
Oh, how the therapy itself, how it works. you
Yeah,
mean?
how the tests work.
Oh, right now it's not in the clinical trial phase. Right now it's in clinic, which means that it's going to be used on patients routinely. But you're asking about clinical trials, how they work?
Yes.
So clinical trials have several phases, and usually clinical trials take years. So you need to test stuff on healthy patients. If you have a drug, you need to test stuff on healthy patients. You need to test doses, and then you test it on actual patients. And in terms of CRISPR, you can't test it on healthy people, honestly, because And so how it was done is that they took patients with sickle cell disease, and they just got their signed approval from them, and they tested how this therapy works on patients already. But before that, it was done on animals. So you already know it has to work on animals, and the animals have to be well in order to move further. But what is really important here is that sickle cell is a fatal disease. There is no cure for it. So moving forward with such clinical trial is easier than moving forward with a clinical trial for some drug for a disease that already has a drug, for example. So when you're trying to cure incurable disease, it's easier to get approval from FDA or from any other agency and go further with it. I don't know if it answered your question.
Thank you.
Okay.
I think there's a lady there behind the gentleman in the white shirt. You wanted to ask a question before, no? No? Maybe it was answered? Okay. I will make you ask questions.
Hi. Thank you very much for sharing your super exciting research. I am curious about nanorobots because I can imagine for like passive drug delivery systems, I can imagine how they find their way through the body. It's like a functionalized like click chemistry or
Yes,
functionalization,
exactly.
maybe like a binding sites, external fields. But can you give me an example, like a basic nanorobot that goes beyond passive drug delivery? Like, how does it work?
Yeah,
I can imagine like it maybe burns glucose to go somewhere.
Okay.
How do you program it to go somewhere?
Yeah, it's a really good question, and I see you are a scientist. Yeah, honestly, right now, many nanorobots are moving chaotically. So you're basically hiring the chance that they're going to meet the correct cell. So our hearts are very powerful. They pump liters of blood in minutes, so the flow is really huge. And passive nanoparticles are just like flowing there. And once you have a nanorobot that is moving at least chaotically, it's capable of like, you know, encountering the correct cell quicker, and the chances are higher. But then you have nanorobots who have directional movement, like you can use a rotating magnetic field for that, for example, and then you can direct them towards the target. So, and also you have nanorobots that are capable of chemotaxis. So for example, when you have a tumor, the tumor secretes inflammatory cytokines or something like that. And those nanorobots are capable to sense it and they go against the concentration gradient of those cytokines into the site of the inflammation, which can be a tumor or anything else that secretes stuff like that.
Okay, so they follow like a chemical gradient.
Yes.
That's already a nanorobot and not a passive system.
Yes.
Okay.
And oftentimes, they also combine several fields together. Like, for example, you have chemotaxis and a rotating magnetic field. And what I really like about nanorobots, once you compare it with passive nanoparticles, the difference is really huge. Even if there is only chemotaxis, it's already boosting the efficiency of the delivery really well.
Makes sense. Thank you very much.
Thank you. And I think a gentleman here wanted to ask a question as well. Over here.
So a quick question. You mentioned that it's already in clinic in the UK. And what is the timeline within the EU and the member states? Is there like a universal approval process for all of EU, or is it member state sort of separate approval?
Well, UK is outside of EU now. So I don't really know what their policies are. And honestly, I don't even know what the EU policies are. It's really outside of the scope of my knowledge, to be honest. But the whole approval was really fast. I remember when those clinical trials started, it was 2021, 2022. It's like a few years ago. It's not really that long ago. And I might be kidding, it might be 2021, but like not earlier than that, as far as I know. So the approval came real fast.
Coming back to the nanorobots, I understand that CRISPR is so sexy that a lot of research groups across the world use it for different, let's say plant discovery, as we heard before, and other areas. What about nanorobots? Is it also such a, let's say, topic which is booming these days? Because let's say in Czech Republic, I know about 2 research groups engaged. So is it really that massive, or is it something much smaller?
I don't think it's as massive as CRISPR, and honestly, I don't think there's anything as massive as CRISPR. Like the scope of how fast it all went from the discovery to clinic is extraordinary. I don't know if we have ever seen anything like it. And nanorobots are a really new addition to the nano family, but it is really booming right now. And especially now, the researchers are communicating with each other a little bit better. Before, we had an issue that material scientists sometimes develop a nanorobot that could never be used in biological systems. So you had nanorobot fueled by something which was toxic. But still, it was a discovery that was important in the beginning. But right now, we know that we need to push those biocompatible fuels and biocompatible materials. So these endeavors really create space for new discoveries. And I'm actually writing a review right now on nanorobotics, and there's so many amazing papers that happened in the last 2 years. And I think it's gonna really change the future, honestly, because once this works, we could do amazing stuff, and also in countries that don't have access to sterile, sterile stuff, like they can't do surgeries or so on. Nanorobots could really change things like that as well.
Thank you very much. I do have a question about controversies now. Give us some juice. Can we talk about some controversial application of CRISPRs, for example? And I'm specifically interested in the dog. In a dog, because I looked at a picture. Alžběta will answer, and I felt sorry for it. But can you tell us what is it about?
Yeah, so CRISPR brought a lot of hope to people who are suffering with diseases. And with this came many, many controversial applications. And there is not that many, but I think even those few are too much right now. So there is a story about a supramuscular dog. I really like to talk about it because it's kind of hilarious and cringe.
It's not a story, it's true, right?
Yeah, it is true. Yeah, it's not like a fairy tale, but yeah. So Chinese scientists came up with this idea to create the supramuscular dog. So in your body, you have a gene that is called myostatin. And this gene produces a protein that basically acts like a stop to your muscles so you don't grow to enormous sizes. So everyone has myostatin. And they basically block the myostatin, the function of myostatin, which means that the dogs that were born had like a huge muscle mass. They looked like a dog Rambo. And back then, it was said that they might use them for like military purposes. I actually Googled like a few months ago and I couldn't find like any more information on that. So I don't know if they're using that. But what is really interesting here and what is really important to note is that this change was done in embryos. So they basically took dog embryos, deleted the myostatin, and created a new dog line of supramuscular dogs. And so the question is, if they can do it in dogs, can they do it in humans? Can we create supramuscular soldiers, for example, like a new line of people that would have super muscles? And editing in embryos is very, very controversial. Back in the day, I say it as if it was like a long time ago. It's not that long time ago. It's 2017, I think, or 18, what we are talking about. But there were no laws that would forbid to do stuff like that. And it continued. So first was the dog, and then Chinese scientists decided to edit human embryos to correct a rare heart disease. But they didn't implant those embryos into a womb, so no humans were born out of that. But it was a step further. It was already in the gray area. There was this question like, should we edit embryos? Like, should we even play with this? And there was another team doing similar thing. And then in 2018, it was this breaking point where there was a conference in Shenzhen, I think, and this Chinese scientist called He Jiankui came up with his research that he actually CRISPRed embryos and let them born. And we have 3 CRISPR babies right now. There are twins, Lulu and Nana, and then there is a 3rd child. I don't know the name of the child, but those children basically cannot contract HIV. So He Jiankui decided to, no one gets why, honestly, but He Jiankui decided that he will CRISPR those embryos, so when they are babies, they cannot get, and adults, they cannot contract HIV. That's the only advantage there. And it was a huge controversy. Like, the scientific community was outraged. People were really scared because the boundary was crossed, and it was crossed really soon. The paper we talked about is 2012, Nobel Prize 2020. This is 2018 when he let those 3 children to be born. So it's really early after the discovery. And what is also controversial about that is not only he created a new line of humans that cannot contract HIV, but also it was discovered that they might have their brains altered. Because there are few people, there were mouse experiments with a similar thing. These mouse had the same mutation that prevented them from getting HIV, and scientists found out that those mice were very intelligent, that they were navigating mazes much better, they had much better memory than regular mice. So it's thought that maybe those children would have this enhancement of better intelligence. But it's not just this intelligence, there's more. So it was discovered that there's also a downside to this. There are several people in the world that have this mutation, that, they are born like that they cannot contract HIV, and they have a huge disadvantages, like they can die much younger when they have multiple sclerosis, or they can die from vaccinations, or like flu and stuff like that. So yeah, and the thing is that those papers were really old. So they were out there even before He Jiankui started this. So it's really hard to believe that he didn't know about them. But he might have decided to ignore them, or it was worth it, even though he, could bring this disadvantage onto those girls. And what is another controversy is that we don't even know if those parents knew about it. If he even told them. And there is another.
Isn't he in jail now for that?
He is out of jail already. Yeah. But there is another thing. Actually, those parents were brought into this idea of He Jiankui in a way that I think is not really ethical. The researchers told them that they could have in vitro fertilization for free if they let them CRISPR their babies, because I think the father had HIV. So it's really, it's a huge, you're taking a chance if you're having a child the natural way, if you are HIV positive. So it's better to have in vitro fertilization, which is really expensive. So they told them, oh, you can have IVF for free, but we will CRISPR your babies. And so this is also unethical by itself. Yeah, and He Jiankui was sent to jail for 3 years, and he's out of jail right now. Yeah.
Well, designer babies and muscular dogs, this is already a fine line. And I think the concept of, you mentioned military personnel with big muscles. I come from a military family. I know muscles aren't what you need if you want to be a good soldier. But the concept of master race, happened globally throughout the history, the past. And I know you told me that you told me that Adolf Hitler got somehow inspired by this therapy or a theory of gene editing. Can you tell us something?
No, not gene editing. No. He got inspired by eugenics, which was born in the US.
Yes.
Many people don't know it. Mm-hmm. It wasn't the Nazis who invented eugenics, it was Americans.
Okay.
So
I thought it was Germans.
Yeah, it was Americans in the early 20th century. They came with this idea that everything is inherited, like your intelligence, your character. Even if you are poor, it's inherited. So actually, the founder of eugenics is Sir Francis Galton, who is the cousin of Darwin. And he came up with eugenics in 1880 first. He coiled this term. And he said that, well, it would be amazing if we could breed out certain characteristics from our genetic pool. And he thought that we could breed character the way we breed color in guinea pigs, for example. Mm-hmm. And of course, he was going after people who were had epilepsy or were promiscuous. He thought all these things are genetic. Mm-hmm. Even like size of a family, genetic, everything. So it was really ridiculous. But then in the early 20th century in the US, they put it into action, and which is really, really scary. And first, it was stuff like eugenic certificates, for example. So if you are considered eugenically clean, you got this certificate, this card, which says that you are capable of eugenic love. You have eugenic love possibilities, or something like that, like some phrase like this. So like when you went on a date, you could show this certificate to your future spouse, like I'm eugenically clean, or like for example, there were these fairs for like where you could show off your stock, like your beautiful cows and your hens with beautiful feathers. And back in the early 20th century, what they did was that they even had like a eugenic house where they showed like the best eugenic families. So it was an exhibition of like cows and stuff, and also eugenic families. And families were competing with each other, which one is more eugenically clean. And those traits that were considered were stuff like jealousness, or the size of a family, or how nice you are, if you're Thai in church, and stuff like that. So it was really ridiculous. And also it was really racist, because all these competitions were, of course, only for white people, because if you were black, you were considered lesser than, and it was like you were not eugenically clean because you were black. So that by itself is very controversial,
Yeah.
and that's how eugenics started. And then it went step further with forced sterilizations, which we are still talking about the US, not about Germany. So forced sterilizations, California actually, where I spent my last year, was the best in sterilizations. They were able to sterilize more people in 1 year than the whole US. And the problem with that, it's a forced sterilization, right? It's a problem by itself, but the bigger problem is that they were sterilizing people like women that were promiscuous, or people with epilepsy, people who were alcoholics, people who were angry, or I don't know, like stuff like that. And the problem is that if you had someone, some relative you didn't like, you could say that he was feeble-minded. And the term feeble-minded, you could hide anything under that. So if the person didn't agree with the government, he was considered feeble-minded and could be sterilized. And there were even institutions which went even further, because it was desirable to get rid of these people. And there were like institutions where you can send people for sterilization, where you can send people to get rid of them. And some of those hospitals or institutions did stuff like they gave them milk with tuberculosis. And they thought, oh, if they drink this milk and they don't die of tuberculosis, this means that they are eugenically clean. Of course they died, which even more reinforced their idea that they were not eugenically clean in the first place. Yeah. And then Germany took an example of it.
This sounds like a good idea for the next Netflix series, which you have just said, but it's something that really happened.
Yes.
Yeah. Alžběta, we are nearing the end. Fast forward from the past to present. What is next for research in this field? And what's next for you as well?
For research, I think the approval of the sickle cell therapy is a huge motivation for the research, and more diseases will follow in the footsteps of sickle cell. So it seems that HIV will be the next, might be the next. So we will be able to finally cure HIV for good in this way. There is amazing research on Huntington's disease. There is amazing research on hereditary deafness. So all these diseases could be next to be approved to be cured by CRISPR, which is absolutely amazing. So, yeah.
Thank you. So what's next for you?
I don't know. Finish my PhD.
Yeah. How is that looking?
Oh, I should be finished soon.
Yeah. Good. Well, congratulations. I have a joke for you on this theme. Before I came down here, I asked ChatGPT to give me a joke on gene editing, because today is 30 years of introduction of ChatGPT in Europe and 30 years in the Czech a year in the Czech Republic. What is Gene Editor's favorite piece of clothing? Designer jeans. Ladies and gentlemen, thank you very much. Alžběta, thank you. All right, let's take some additional questions. If you have any after what has just been said, anyone would like to ask anything? No, let's save it for networking, shall we? Okay, well, thank you. Thank you very much.
First of all, I would like to thank to all of you for coming tonight and for being solidar and wearing a face mask tonight. And of course,
That was because of me, and I'm really grateful for that. Thank you.
indeed, thanks a lot for that. And I cannot forget to thank both of you on the stage for navigating us for at least for so many of us still a very difficult topic. Thanks a lot for that.
Thank you, Daniela. And now I would like to invite you downstairs. There is a really nice exhibition of Jiří Peterbok. And also, as usual, I would like to invite to the glass of South Moravian wine. And thank you very much also to all of us, to every team of Science Café. And thank you very much. And see you soon.